You know, the difference between a solid B and an A star in Cambridge Biology 9700 often comes down to one really specific skill: totally mastering the eyepiece graticule and stage micrometer. I'm going to show you exactly what examiners are hunting for and how to lock in those top-tier marks by treating this whole topic not just as some dry math problem, but as a core biological principle. Picture this. You're sitting in the exam hall. You've aligned your scales perfectly. You've counted the divisions, you've done the multiplication, and your final number is mathematically flawless. But when the results come out, you look at this exact question and realize you scored nothing. How does a mathematically perfect graticule calculation score exactly 0 marks? Honestly, it's the ultimate exam nightmare, and it happens to incredibly smart candidates every single year. 0 marks. Ouch, right? Let's dive into this super costly mistake. It is absolutely devastating to lose points on a calculation you actually conceptually understand, all because of a forgotten rule. Countless students get 0 marks simply by forgetting a really basic foundational rule about how Cambridge marks these specific questions. Well, today, we're fixing that completely. We're going to build a mental framework that ensures you never, ever leave these easy marks on the table again.
All right, Section 1, Core Principles and Terminology.
So to actually beat this exam, we kind of have to step away from the math for a quick second and establish the theoretical architecture that Cambridge examiners expect to see on your paper. According to the Principal Examiner Report, candidates referencing cross-topic connections achieve significantly higher marks. What does that mean for you? It means examiners prioritize analytical depth and conceptual understanding way over just superficially memorizing a step-by-step methodology. They want to see that you truly understand why you're calibrating that eyepiece graticule with the stage micrometer rather than just how to plug numbers into a formula. And this brings us to conceptual understanding. Top-performing students, they don't just calculate, they successfully link this tool to totally different areas of the syllabus. The exam might actually ask you to apply your graticule measurements to a completely unfamiliar biological context, maybe a unique tissue sample or some novel experiment you've never seen before. If you've only memorized the math, you're going to freeze. But if you have true conceptual understanding, you can transfer that logic literally anywhere. Moving right along, let's look at Section 2, the method mark strategy. So how do we build a foolproof procedural framework to secure those guaranteed method marks, even if your final answer somehow goes a little off track? In a minute, I'm going to share the exact examiner clue that completely changes your answer. But first, you have to strictly follow this 5-step standardized sequence. 1. Identify the key elements, meaning spot the stage micrometer value and the eyepiece divisions. 2. Apply the relevant principles of calibration. 3. Execute your analysis with crystal clear working. 4. Verify your results so you know they actually make biological sense. And 5, present it in the appropriate format. Look, if you skip showing your working because you just did the math in your head, you lose the method marks entirely. Write everything down. Now, because interpreting your data is just as critical as gathering it, you've got to extract information precisely and match the correct statistical analysis test to your graticule data. Say the question asks you to evaluate the size difference between 2 sets of cells you just measured. You need to know which test applies, right? The t-test is for comparing means, chi-squared is for categorical data, and correlation tests, well, those are for finding relationships between variables. Matching your practical measurements to the correct statistical tool is a massive marker of an A-star student. And this perfectly illustrates the ultimate problem-solving framework for Biology 9700. Step 1 is to deconstruct. Identify the given information and break the whole thing into logical steps. Step 2 is solve. You systematically work your way through the math. Step 3 is evaluate. Check the reasonableness of your answer and justify your recommendations. Think about it. Did you just calculate a single red blood cell to be 3 centimeters wide? Because if you did, your evaluation step should immediately set off alarm bells that a decimal point is definitely in the wrong place. Okay, let's jump into Section 3, flawed versus flawless answers. Let's peek right into the examiner's playbook. If you contrast a typical flawed student answer with an elite top-band flawless response, the difference is honestly night and day. The flawed approach relies on really vague reasoning, totally generic numbers, and crucially, forgets to state the units. Remember that hook question we started with? How to get 0 marks on perfect math? It's right there, forgetting your units. On the flip side, the flawless answer identifies both similarities and differences using clear comparative connectives. It supports every single claim with specific evidence from the data, and precise units are included at every single step of the way. And that leads us straight to a massive examiner alert. Always link every point directly to the specific scenario context you've been given. This is such a classic mistake. Providing generic textbook answers that aren't tied to the actual experimental context in your paper will completely cost you the grade boundary. If your question is about measuring palisade mesophyll cells in a drought-stressed leaf, your answer absolutely cannot just be about calibrating a microscope in general. It has to specifically reference those mesophyll cells and that exact environmental context. Which brings us to Section 4, Top Examiner Warnings. Straight from the Cambridge Principal Examiner reports, let's break down the major pitfalls that trip up even the smartest, most prepared candidates. Keep this little checklist running in your head during the entire exam. Number 1, never be vague. Examiners demand specific, granular details. Number 2, religiously check your units and terminology. A micrometer is vastly different from a millimeter, guys. And number 3, always apply your methodology directly to the specific practical work scenario. If you just check these 3 boxes before turning the page of your exam, you are actively protecting your marks. Finally, Section 5, the final challenge. Before we wrap up this explainer, I want to leave you with one last high-stakes scenario. It's a trap that catches almost everyone off guard, even if they've memorized the methodology flawlessly. If your Graticule math is 100% correct and your units are absolutely spot on, what hidden trap could still drop you to a lower mark band? Really think about it. You followed the entire framework perfectly, but you still lost the points. Why? Because you completely misinterpreted the examiner's command word for the question. You described when you were actually supposed to explain, or you calculated when you were supposed to evaluate. Command words. They are literally the ultimate gatekeepers of the mark scheme. So the very next time you face a Graticule calculation, ask yourself, are you just crunching the numbers, or are you actually answering the examiner's specific command? To solve this exact problem and secure those final crucial marks, subscribe to our YouTube channel Ace the Exams at acetheexams_IGCSE to excel in your Cambridge examinations, and immediately launch our next explainer on decoding command words. We'll see you there.